Welcome to the deep dive. Today, we're diving into how human cells export their messenger RNA. And honestly, the single biggest point, it boils down to this one protein, an ATPase called UAP56. We knew it was there, but now we know it's basically the master switch, using ATP to control the whole process, from packaging right up to release at the nuclear pore.
Yeah, it's a really fundamental process. For genes to work, the mRNA blueprint has to get out of the nucleus. It gets packaged into this thing called an mRNP complex, and then it has to squeeze through the nuclear pore complex, the NPC.
Okay.
We knew the loading machinery, the TREX complex, involved the THO complex, and as you said, UAP56. But how UAP56 actually managed the handover for export, that was, well, a bit of a mystery.
Right, so let's break it down. First, big question. How does that initial TREX complex, which is holding the mRNA, how does it actually let go so the mRNP can move? How does it remodel?
Okay, so this is where UAP56's motor function, its clampase activity, really shines. It needs two things: ATP for energy and the RNA itself to grab onto.
Right.
When UAP56 uses that ATP to clamp down hard on the mRNA, its own shape changes. And researchers showed this directly using gradient-coupled interferometry. They found that while the THO complex normally binds UAP56 pretty well, that binding just vanishes, completely disappears, when UAP56 is in that ATP-fueled, RNA-clamped state.
Okay. So the very act of UAP56 clamping the RNA provides the force, like a kinetic lever, to push the original TREX complex away. It sort of kicks itself off the loading dock.
Exactly that. It uses its own action to initiate the separation. But, okay, think about this. Now you have this UAP56 mRNP complex floating free. These kinds of multi-part complexes can be pretty unstable. So what stops it from just falling apart or, drifting back to the HO?
Good question. What does stabilize it for the journey?
Well, that's where another protein, SARMA, comes into the picture. They found it using a computational screen, actually. And SARMP is interesting because it had these 5 specific motifs called UAP56 clamping motifs, or UCM.
5 of them.
Yeah, 5. And they work together synergistically to grab onto that UAP56 while it's clamped to the RNA. Think of it like multiple safety clips holding everything secure.
Okay, so it reinforces the clamp.
Precisely. And in experiments where they watch TREX disassemble, adding just little bits of SARMP, these UAP56 significantly boosted the release of stable UAP56 mRNPs from THO. SARM basically acts like a chaperone, making sure UAP56 stays clamped and committed to export.
All right, so the mRNP is packaged, stabilized by SARMP. Now it needs to get through the gate, the NPC. It docks using another complex there, TREX2. But if UAP56 is still clamped tight onto the RNA, how does it finally let go? What's the release mechanism?
This was a key piece of the puzzle. They found the unclampase, and it's actually part of TREX2 itself. Specifically, a component called GNMP.
GNMP?
Yes. Using cryo-EM, they saw this conserved loop within GNP, which they nicknamed the wedge. And crucially, this wedge sits right next to where UAP56 binds ATP.
Oh, interesting. Like it's poised to interfere.
Exactly. It's not just a docking platform. TREX2 actively stimulates UAP56's ATPase activity, makes it burn through ATP much faster. We're talking over 50 times faster when RNA is present.
Wow, 50-fold.
Yeah. And this massive boost in activity, likely triggered by that JMP wedge physically poking the ATP site, seems to be the trigger. It forces UAP56 to finally unclamp, releasing the mRNA cargo so it can be handed off to the actual export factors, NXF1, NXD1, and pass through the pore.
Okay, so this really paints a full picture then, a kind of 5-step assembly line. Load with THO, clamp with UAP56 ATP, causing THO release, stabilize with SARMPO, dock with TREX2, and then TREX2 itself triggers the release. UAP56 is absolutely central.
Absolutely. It connects the loading phase with THO to the unloading phase with TX2, all controlled by its ATP cycle, and the implications are quite broad. Well, this whole mechanism, using an ATPase clamp that's then regulated by other factors, looks remarkably similar to how another key RNA complex, the exon junction complex, or EJC, is handled by its ATPase, EIF4A3. It suggests this might be a common strategy, a conserved playbook the cell uses for controlling different RNA processes.
A repeating pattern.
Exactly. Plus, it gives us a solid mechanical basis for this idea called gene gating. The observation that genes being actively transcribed often seem to be physically close to the nuclear pores. This whole UAP56 pathway makes that localization make perfect sense for efficient export.
Fascinating. Okay, so let's quickly recap the 3 main takeaways for everyone listening. First, UAP56 clamping onto mRNA using ATP is the force that breaks apart the initial TREX complex.
Right. Second, SARMP jumps in to stabilize that newly formed UAP56 mRNP, keeping it ready for export.
And third, the TREX-2 complex at the nuclear pore doesn't just dock the cargo. Its JMP wedge actively simulates UAP56 to unclamp and release the mRNA right at the exit.
Perfect summary. Though it's worth mentioning, one small caveat perhaps is that SARMP's role might have some backup. The authors suggest other factors could potentially help stabilize the mRNP too.
Always more layers to uncover.
Always. But it leaves you with a really interesting thought. If this UAP56 clampus mechanism is mirrored in the EJC pathway, what other RNA helicases and protein complexes out there might be using this same kind of regulated clamping strategy? It makes you wonder where else this principle applies in the cell. Something to think about.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.